What we're going to do is take a series of images. It's called a fly-through, and you're going to start at the surface epithelium. We're going to go into the cornea, and you're going to see these little round dots. They're going to be similar to the black dots that you saw in the histologic section that actually represent the nuclei of the keratocites. If this movie plays, hopefully. This is from a living eye, and this is from obviously a fixed and processed eye. We can look at individual slices, and that's shown here. This is that confocal image showing these nuclei. And here's a histologic image taken in the same plane showing the cells. So the nuclei are here, and you can see the cell body. So there's quite a bit of cell body. And this is where the cystine crystals reside, inside the body, inside the lysosomes that you've already heard today. So what we wanted to do, this is just some different images, but what we've used is this imaging modality to look at the natural history in the CTNS mouse. You can see here some crystals forming, and we've looked at a series of mice, both control and 14 cystinosis mice, looked at them at various times sequentially over age, and then tried to monitor the amount of cystine that develops. I'll first show some of the images that we get from these mouse corneas, then followed by the quantitative analysis. Now early on in the young mouse, 3 months, we first begin to detect some cystine crystals. You can see the slight crystal right here that's within the corneal endothelium in the very back layer of the cornea. As you move on in age, these crystals then get larger and get quite prominent. They get larger than the size of a cell. You can begin to see crystals in the stroma. As you can see here, here's the next Z slice. This would be the epithelium. This is the endothelium, and we see all these crystals in the corneal stroma that develop, and this is what's scattering all the light. Now we've gone on to quantify this using an image processing program, using digital imaging software, in which we can segment out the crystals individually, and that's shown here in a three-dimensional rendering. What we do essentially, and I don't know if you're going to be able to see this either in this excessive light, but this is the optical slice around the epithelium, and as we go in, we're going to see those crystals pop up. We come back to the endothelium, and you're going to see a lot of crystals in the endothelium. Again, stromal crystals are going to show here. Then when we segment out, we'll specifically identify only the crystals. This is this yellow. And then back to the endothelium. And now we can pop out just the crystals alone. And now we have a measure then of the volume of the crystals within inside the corneal stroma. Since we're looking at the same animal over time, we can make these same measurements and see how crystals deposit over time and then look at the effects of therapy. And that's what we've sort of done here, looking at the temporal sequence of crystal load in a series of mice. You see, we first detect crystals at 3 months. They increase in number and size up to about 7 months, and then they appear to drop off over time. Yes?
On that computer-generated image, there were large, are those clusters of crystals? Or
there are clusters of crystals in the back.
Okay. Do they grow together, and does it take time to act to
we haven't seen that, but what happens, I think, over the process is the cells die. They actually, the inflammatory cells come in and begin to break down, but since they can't process the cystine very fine spicule-like crystals get broken down into sort of clumps as part of the process. And if you had the inflammatory cells that could handle the cystine or transport it, then those crystals would leave. But we're getting a decrease over time in the crystal load. And I think that's sort of explained as we begin to look at the pathophysiology. So we've looked at eyes sequentially at different time points by sacrificing the animal, taking out the corneas, and looking at the cells and what's going on with the cells. And these are images, immunocytochemistry. Now these slices then are similar in orientation to what you saw with the in vivo confocal images, and where you see red nuclei, and then the green here is actin bordering the cells. The cells are these very large dendritic cells. There's a lot of cells, and they all reach out and touch each other. And at 2 months, they're pretty normally organized. But at 5 months, you begin to see some very intense staining of actin, some fragmentation of nuclei, and this intense staining of actin is really associated with an infiltration of inflammatory cells. And more inflammatory cells infiltrate at 10 months, and there's fewer cells and more abnormal keratocites. And by 12 months, we can actually see regions where the keratocites are dropping out. So what we think is happening is that as these cells begin to deposit these large crystals, the crystals are injuring the cell, ultimately killing the cell, causing inflammatory cells to migrate in. You get fewer cells over time. The crystals then are phagocytosed and then probably removed from the cornea, but still new cells are there, and they're collecting more crystals, and you have more light scattering. This is just to show that these cells that migrate in with this very bright green actin staining are actually inflammatory cells. This is a marker, CD45, for acute inflammatory cells. And you can see it lights up very nicely with these cells that are infiltrating at these different ages. So that part of the process of this disease then is damage to these very important keratocytes, cell death, migration of inflammatory cells in, and this can ultimately lead to neovascularization and scarring, which occurs in the mouse around 7 days. The same kinds of changes are also occurring in the endothelial cells, as you can see there. So what we think is happening as the natural history in corneal cystinosis, you get this increasing cystine crystal load. This begins to cause keratocyte cell death, which increases over time, and you get loss of cells. As they die, you recruit inflammatory cells come in, and ultimately you lead to a neovascularization and scarring of the cornea. But our goal has been to replace these CTNS minus corneal keratocytes then with CTNS plus plus. And Dr. Shaw already presented one approach, gene therapy. There's also been the approach of using bone marrow stem cell transplantation. I'll talk a little bit about that. And then also a new approach there's 2 papers recently out on keratocyte progenitor cell transplantation that I've had the opportunity to work with Dr. Cow on that also may be a promising approach. So bone marrow transplantation, we can take cells from a green mouse. These are cells that express GFP, which is a green fluorescent protein. We can take these cells and inject them into the CTNS mouse or any mouse that's a normal mouse that doesn't have green cells. Usually you have to lethally irradiate them so you can get the cells to implant. Then you can record what's your reconstitution rate, and we get a pretty high reconstitution rate. This is an image from Dr. Cherky's paper that we collaborated with, looking at the green cells that migrate into the cornea. You get these little processes sticking out. Some cells here, what we believe are all inflammatory cells that are being recruited due to the dying of the keratocytes. And you can see here this arrow points to a vessel that's out of the periphery of the cornea, and you see all these green cells that have migrated in. We've gone on and shown that all the green cells, that when you transplant green bone marrow into a normal mouse, the green cells line up very nicely with this other marker for immune inflammatory cells, CD11C, which is actually a marker of macrophages. You can see all the yellow here relates to the fact that there's a pretty good reconstitution of all of the macrophages and dendritic cells with these green cells. So we've also gone on and tried to look at what these cells are doing. We take strips of mouse cornea, we put it onto a in vitro device where we can suspend it and then look up with a microscope and take 3-dimensional images through the cornea, as you can see there in this mount. And we use a special kind of imaging approach using multiphoton to actually collect 3-dimensional time-lapse images, and that's going to be shown here. And if Brian would be so kind, what you're going to see here is there's an XY plane, an XY plane and an XZ plane, and we'll want to play this again. And these are actually the cells, the green cells that have migrated into the cornea, and you can see they're migrating quite fast, and they migrate all over. We want to play that again. This happens to be an XZ slice, and you'll see a cell zoom around here, dive, move. So these cells are quite active, and they're all part, we can go back to the other one, all part of an immune surveillance type mechanism that's going on in the cornea. So when you do bone marrow transplantations, these cells end up in the cornea, and they're doing immune surveillance. They're looking for cells that are dying and that they're going to phagocytose. Then they will be able to process the cystine crystals. We've gone on and looked at the response of these types of transplanted mice, continue on, and shown that these cells, after injury, would migrate into the cornea, but none of them stay around very long. After about 14 days, they leave, and you just have a few residual, and we've gone so far as out to 9 months, and never seen any of these cells become keratocytes, and we need to have the keratocytes replaced. because these are the cells that keep the cornea transparent. So we moved on to another approach looking at keratinocyte progenitor cells, and there's two recent reports, one in stem cell and one in +1, one by a group in Pittsburgh, Dr. Funderburg's lab, in which he has isolated a progenitor cell from the cornea of humans and shown that these cells can be injected into mouse corneas and that they will replace some of the keratinocytes that are present there and correct a genetic disease, Lumican deficiency, which is an extracellular matrix disease. In addition, Winston Cow has gone on and identified that you can also use umbilical mesenchymal stem cells. Now, mesenchymal stem cells are, again, a progenitor type cell that can differentiate into different types of cells, such as connective tissue cells, bone, fat, as well as cartilage. Let's move on. So we've then taken this approach, looked at these progenitor cells and transplanted them to mice. This is a mouse cornea, and this is a needle sticking into this interstromal space, and we inject these multipotent stem cells that can differentiate into various stem cell into various tissue types, and gone on and looked to see if they can differentiate to keratinocytes. So here's some labeled cells, umbilical mesenchymal stem cells that were injected in the cornea. This is the bright field showing the edge of the cornea. This is cornea over here. This would be sclera over here. And you can see these green cells are all present within the corneal region. If you take a histologic section and look at the green cells, here they are. Here's the surface epithelium on the cornea. Down here's the endothelium. These are the stromal cells. We also have a marker of inflammatory cells. You do get a little bit of inflammation when you do inject these cells. But when you look at what happens to the cells over time, initially you get a bolus of cells in the cornea, and then they begin to spread out, and then they fully fill the cornea, and they begin initially as sort of round cells, like the cells they are initially when you inject them, and then they also spread out and appear to become more dendritic. And if you look here, here's a high power magnification. This is a dendritic keratinocyte, and it's labeled with this red dye, indicating that this was one of the cells that was initially injected. It was a mesenchymal stem cell that now has become a keratinocyte. In fact, we've gone on and shown that these cells express markers of keratinocytes. They express the correct proteins, extracellular matrix molecules that's important for keeping the cornea transparent. So we've gone on, we have a preliminary study. We've only got 10 weeks out. We don't even have a quantification, but I wanted to present this. We've injected 8 mice. One eye was injected with saline, the other eye was injected with 10 to the 4th umbilical mesenchymal stem cells in a volume of about 2 microliters. It's very small. The other eye was injected with saline, and we looked at these at 10 weeks. These are in vivo confocal images. The left is the saline injected eye, the right is the umbilical cord mesenchymal stem cell in this same mouse. So if we look here, we see stromal and then endothelial crystals, that's the iris. Stromal and endothelial. And then if we look at the saline, there's no stromal, but there is some endothelial, which we'd expect because these cells can't get back to the endothelium. So we have an absence of stromal crystals in this particular mouse. So we see no stromal crystals, and yet there are crystals in the endothelium. Here's 3 different mice. We can see here's all the saline injected eyes with crystals, and here's the eye injected with 3 different eyes with the umbilical cord mesenchymal stem cells. Also interestingly, 3 out of 8 eyes injected with saline have gone on to fail and develop scars, where 0 of the 7 eyes injected with umbilical cord mesenchymal stem cells have not shown any scarring. So in conclusion, we believe that the CTNS mouse develops a corneal cystinosis that's very similar to the clinical condition. You get accumulation of these cystine crystals in keratinocytes that perhaps lead to keratinocyte cell death, stromal inflammation, leading to scarring and neovascularization. And that with interstromal transplantation of umbilical mesenchymal stem cell may slow this progression by replacing those dead cells or those dying keratinocytes with actual functional keratinocytes. And with that, I'd like to thank you. I'd like to thank the Cystinosis Research Foundation for funding this research.
Dr. Judy.
If you irritate the surface of the eye, so the epithelium rough up a little bit, some lemon juice, or I don't know how it would be done, but if you were to irritate the epithelium and then apply progenitor cells directly to the surface, would they enter the stroma?
That's probably not likely in the human. The human cornea has a unique, what's called a anterior limiting lamina that's about 10 microns thick. It's a very tightly woven collagen network. It's difficult for cells to migrate through that. But with the refractive surgery techniques we have today, with the ability to form tunnels inside corneas for doing LASIK surgery, I'm sure all of you are familiar with the refractive surgery that's available today. They can make tunnels to be able to inject material into the cornea fairly easily. So that's not really a problem. You wouldn't have to stick a needle in a patient. You could actually very precisely with these lasers today make tunnels to do that. Oh,
I noticed that there seems to be a tendency for cells that are injected into the cornea to move through the cornea, through the depth of the cornea, and then disappear, go away. What causes the cystine crystals to persist in the corneas?
well, the cystine crystals actually form inside the resident cells, stromal cells. The resident stromal cells or the keratocytes do not move. They are quiescent sitting. That's a problem with gene therapy. These cells don't divide. So you're not going to get any integration of genes into these cells. That's the problem with bone marrow, and that bone marrow cells sort of migrate through and they're moving around. But the stem cells that we're using, these progenitor cells, then differentiate to the keratocyte and then sit there and reside and don't move.
So given that the crystal is actually inside a dead cell, it's killed the cell because it's crystallized, is there any way to trick cells that move through to tell them that this isn't a resident cell or resident residue of a cell so that they can transport it out?
Well, until the cell dies, it's sitting there with these crystals and it's not giving any signals to recruit cells. When the cell begins to die, then signals are released to recruit these inflammatory cells in. And then they phagocytose, digest the crystals, and if they have the gene, then they can process it. Otherwise, they sit there and will phagocytose and stay, or a lot of times leave. And I think that's probably explains why we're seeing this decrease in crystal over time after 7 months. The cells are being digested and then they're being removed. Yes?
So I'm just interested in the history of the formation. What happens in utero? It seems like at 3 months there's not many crystals and then they increase. Why are there not more crystals when they're born? Is something in utero which is protected?
Why there's this length of time before you get crystal formation and this continued crystal formation, I have no clue. I would have to leave that for Dr. Schneider or one of the other ones. But you don't detect it immediately. Sometime around 3 months in the cornea you detect it, and I think that's also true in some of the other tissues. It takes some time for these crystals to begin to form. And you can take these cells out and grow them in tissue culture and they don't form crystals. So, I don't know. Yes, ma'am.
It sounds, this is really exciting because you've had some exciting results in the last couple weeks. So, as a mom now speaking and wanting to know when maybe there's something that we call proof of concept, or how much more time do you need to keep measuring these kind of hopefully successes?
That's a good question. Well, I think first of all, we're presenting 10-week data and we'd like to see 1-year data to be sure what we're doing. We'd like to know when is a good time to inject. Can you inject when you have full-blown crystal formation and you start getting devascularization, or is this something that needs to be done early? And then what happens when you injure? Do these cells that we're putting in there, do they stay as keratocytes and repopulate as keratocytes, or do they become some kind of scar tissue? Those, I think, questions need to be addressed first. But then again, injecting these kinds of cells in the cornea is somewhat of an immune-privileged site. So you do not get a lot of immune response to these kinds of cells, and stem cells are somewhat, these umbilical mesenchymal stem cells lack some of the markers that would be seen as rejection, and it's already in an immune privileged site, so I think it's a possibility to use these in that kind of a setting. although there's also the IPS, but then we need to know how to get the IPS cells to be able to differentiate to their appropriate cell. And that hasn't been done yet. So, but I don't know. Year, 3 years, 5 years.
Thanks, Dr. Jester. Isn't it nice to know for us as families and donors, you know, that every dollar that we raise, whether it's $1 or $1 million or whatever the denomination or however it's raised, is going to these mines to do what we're doing. I mean, I think it's awesome, comforting for us as families, and also when we're out trying to get people to, you know, to donate. I mean, this is what's going on as a direct result of the CRF and the doctors, so it's great.